Okay, so we are going over the problems to genetic problem set 1. Alright, so number 1 asks you if pure breeding. Pure breeding is the same as true breeding. So if you're crossing a true breeding pea plant with a true breeding short pea plant, what will be the genotype and phenotype ratios of the offspring? Tallness is dominant. So tall is dominant, short is recessive. So your cross is true breeding, which is big R, true breeding tall, cross with true breeding short, which is little r. So you do the cross. This parent could pass on a big R or a big R. This parent could pass on little r or a little r. And so we get all of our kids being big R, little r. So our genotype ratio would be 0 big R to all 4 being big R, 0 little r. Your phenotype ratio would be then all 4 of them are tall to 0 short. Whoops, I'm off this page here. To 0 short there. Alright, so that is number 1. Number 2 asks you if the offspring of the above cross are crossed with itself, what will be the genotype and phenotype ratio? That's these guys here. So the offspring are the big R, little r. So your cross is big R, little r, cross with big R, little r. So you set up your Punnett square. This parent could pass on the big R or the little r. Same thing with the other parent. So your Punnett square looks like this. And so your genotype ratio would be 1 big R to 2 big R, little r's to 1 little r. Your phenotype ratio then would be these three tall to 1 short. So number 3 asks you if 500 plants were produced by the F1, so that's these guys right here are the F1 generation. So the plants that were produced are these guys here. If 500 plants were produced, how many would you expect to be short? Alright, so let's look here. We'd expect 1 out of 4 to be short, right? And so if they had a total of 500 plants, you'd expect 1/4 of them to be short. So what's 1/4 of 500 is 125. Number 4, a black hog is a male pig, was crossed with a black sow. Sow is a female pig and producing 12 pigs total. So we're black crossed with white. It gives you 12 pigs. 9 pigs were black and 3 pigs were white. So that's a 9 black to 3 white to give us a total of 12 kids from this cross. That's your ratio of the kids. And it tells you black is dominant. So black is dominant to white. What is the genotype ratio of the offspring? When we look at the cross, that would give us results like this. If you notice here, 9 and 3, just like I was talking about with Mendel's P's in taking the smaller number and reducing it by taking the smallest number and dividing it into both numbers here. If we reduce this by 3, this is a 3-to-1 ratio. So what kind of cross would give us a 3-to-1 ratio? And that, so that we're kind of working backwards. So these guys have to have little b's, and these guys have to have a big B. So to have a little b here, to have little b's, you have to have each parent have to have a little b. All right, and so that would give you a little b kid. But then the 3-to-1 ratio, all of these would have to have a big B, which means then that the parent has to be heterozygous. So if we get a heterozygous parent like this, all right, this is what the cross looks like. And so your genotype, the question is asking about genotype ratio, so we'd expect a ratio like this, 1 to 2 to 1. All right, that's the reduced ratio. If we make it, so this would equals 4 kids, if they had 4 kids, they had 12 kids. So if we multiply this by 3, this is really what their ratio would look like. 3 to 6 to 3. That's the same as a 1 to 2 to 1 ratio and gives them a total of 12 kids. So either one of those is fine, because if you reduce it, that's the same ratio. So 5 asks you what term describes the hog and sow's genotype. The hog and sow were these guys that you crossed here, so what term describes them? They're both heterozygous. All right, number 6, if you choose a black pig from this litter for breeding, what are your chances that it would produce only black offspring if crossed with a white sow? So, the type of black pig, so you have 2 different kinds of black pigs. You have these 2 and you have this one. And so which one of these, if you cross it with a white sow, the white sow is going to be little b, little b. So when you do a cross here and you're crossing with a white sow, which kind of pig, black pig, big B, big B, or big B, little b, would give you only black offspring all the time? And that would be the little b, big B, big B. If you picked a big B, little b, then you would get these 2 right here. If this was a little b, you'd get really 50%. You'd expect to be white. So we're looking for a black sow that has big B, so that you 100% of the time, all the time, get a black sow. And so what are your chances of picking a big B pig from this litter? So like you have the 12 litters, or 12 in your litter, and what are your chances of picking a big B pig? And so one might think 3 out of 12 or 1 out of 4 or 25%, but really when you're looking at your litter and you're just saying, okay, I want to pick a black pig that's going to give me only black offspring, I'm not going to consider these guys because these guys are white. So and I could see that they're not black. So I'm only going to choose out of these guys, and these guys all look the same. So what is my chances? It's 1 out of 3. All right, so because I'm not going to choose these guys, so out of 3, 1 out of 3 would be big B, and I have a 1 out of 3 chance of picking that pig. All right, 7. 7 is dealing with incomplete dominance. So you have a white petunia crossed with a red, and all the offspring are pink. So this is the blending. Red crossed with white equals pink. They give you pink. Show the Punnett square for this cross. So the red is going to have 2 red genes. The white is going to have 2 white genes. So all the offspring are going to be heterozygous. And so all of them have a red and a white gene, so therefore all of them are pink. So that's the Punnett square. Number 8, what conclusions can be drawn regarding the parent plants? Because they show incomplete dominance, we knew without a doubt that the parent plants had to be homozygous. And so the red parent had 2 red genes, and the white parent had 2 white genes. So they both have to be homozygous. All right, number 9. Now cross your offspring from question 7 with itself and show the Punnett square. So I'm going to do that over here. So the offspring are the pink parent plants. So you're crossing a pink, which has a red and a white gene. All right, so that's the cross there. And show the Punnett square. So that was a number 9. Number 10 asks you if 360 flowers were produced, how many would you expect of each genotype? So the genotype ratio, if we do the ratio, would be 1 of this, 2 CR-CW, 1 CW-CW. So that's your genotype ratio, but it's asking you if 360 flowers were produced. So we want to, this is number 10, so we want to change this to represent a total of 360. So you'd expect 1 out of 4 to be this. So what's 1/4 of 360? So you'd expect 90. And then 2 out of 4, 1/2, or 180. And 1 out of 4, or 1/4 of 360 is 90. So it's going to be your 90 to 180 to 90. So those are your actual numbers that you'd expect. So number 11 asks you, can you tell the genotype for this trait of a petunia just by looking at it? And that is absolutely true. So looking, you can tell both the homozygous, and if it's heterozygous, it looks different than either one of the other two because it's a blending and it's pink. So for incomplete dominance, we for sure know the genotypes of everybody. So that's number 11. So number 12 asks you if pink and white petunias are crossed to produce 400 seeds. So pink with white. So pink is going to be CR-CW. White is CW-CW. So this is what that cross looks like. So you fill the Punnett square in. This one will have CR-CW, CW-CW, CR-CW, CW-CW. All right, so and asks you what percent will be red flowers after they've grown and matured. This is 0% red. It's impossible for them to have red flowers. So then 13 asks you what percent is expected to be pink. So if we look at this, I guess I can write that up here for 13. You'd expect 50% to be pink. That's 13. And 14 is what is expected to be white. These two are white. And so you'd expect 50% to be white. All right, so then number 15. Number 15 is a difficult kind of question. It says there are some male flies that show no interest in mating with females. What will happen to the allele over a long period of time? Would it make a difference if the gene were dominant or recessive? And explain. So let's go through some scenarios. So let's say the big R is no mating. So it's a gene for no mating. And little r is a gene for mating. So you have male flies, and there's 3 different genotypes. They could be big R, little r, or little r, little r. And so in this case here, if we look at this, no mating, these guys, the big R, these are not going to mate, right? So this is a no mating gene. These guys will mate. These guys, if they mate, they're only going to pass on a little r to their kids, all right? And so they'll pass on the little r, but the no mating gene, if it's a dominant gene, will never be passed on if it's dominant, and therefore, what will happen to that? If it's a dominant gene, it will die out. It will go down over a long period of time because this, and eventually probably die out because anybody who has this dominant gene is not going to pass it on to the next generation. Versus if the no mating gene is recessive and the mating gene is dominant, then we have still three scenarios. And these guys here, these guys are the ones that aren't going to be passing their genes on to the next generation. These guys will, so these guys will be mating. notice that this one is carrying that no mating gene. So is it possible for this one to pass that no mating gene on to their kids, and this no mating gene, stay in the population for a while? Absolutely. It can be hidden in the population for a long time, and it's not until maybe the female is carrying that this one mates with that's carrying it, and then you get a no mating fly. But so it can be hidden in the population. So the answer to this question is the allele can go down over time, with either one of them. If it's dominant, though, that allele most likely will be wiped out. If it's recessive, it most likely won't be wiped out, because people can be big R, little r, and carrying that gene without showing it, so it can stay around in the population. And this is how a lot of recessive genetic diseases stay around in the population, and people are big R, little r for these diseases, even though they may be horrible diseases. But it can stay around the population because it's recessive and can be hidden for many generations and then show up later. So whether it's dominant or recessive makes a difference with that one. All right, so then number 16. Get a new piece of paper here. Farmer Brown crossed the white hen with a black rooster. He produced 100 chicks. And he got 50-50 ratio. 50 white to 50 black. And it asks you if one of the genes is dominant. So what kind of cross would give you 50 white and 50 black? So looking at this, you get white or black, crossing a white and a black, to get a 50-50 ratio. One of them probably is dominant, because there's no indication of blending, because if white and a black would give you an intermediate of gray. And so if it was incomplete dominance, this one would have 2 white genes, this would have 2 black genes, and then all the kids would have a white and a black gene, which would make them gray. We don't see that, so therefore, in this particular case, one has to be dominant, one has to be recessive, white or black. So we don't know what's dominant or recessive right now, white or black, and so one of them has to be little b or little r, to be recessive. And so one of them would have to be that, to have also recessive kids. They have to have a recessive kid. One of them's black and one of them's white, and so therefore, to have a recessive kid, you have to have a parent that has at least one recessive gene. And so, but the parent shows the other trait too. It shows, one of them shows the dominant trait, one shows the recessive. And so it's a cross like this with a big R that would give you the outcome that you would want in your kids. 50% one, 50% the other. In this particular case, so we know what the parents are. This is the genotypes of the parents. So the answer to the first part of this is one of the genes dominant is yes, and explain. We already did that, and so I guess we already answered number 16, that one of the genes has to be dominant because the kids are white and black and not gray. And so number 17 asks you, if so, can you tell which one? Now, can you tell if this is the white or the black parent, and this is the white or the black parent? The answer is no, based upon the information that they give you. So this could be black and this could be white, and you'd get 50% black, 50% white. Or this could be the white and this could be the black, and you get 50% black, 50% white. So it doesn't really matter. There's nothing you can, from this information, that we can tell. So number 18, 17 was no, you can't tell which one. 18, how are both parents homozygous? And that is no. One parent has to be heterozygous and one is homozygous recessive in order to get that 50-50 ratio. If both parents were homozygous, all the kids would have the same genotype and phenotype. And so number 19, what are the possible genotypes of the parents? We already did that. One is big R, little r, one is little r, but we can't tell which one is which because it has to be certain to tell which is which, and we can't based upon the information given to us. All right, and then the last thing, number 20, if you knew the genotype of one parent, could you determine the genotype of the other and explain? So for number 20, if you knew, so they're saying here in the Punnett square, if you knew the genotype of one parent, so you knew one parent, let's say, like a lot of times you know the recessive because because recessive has to have two lowercase letters, two recessive genes. And then another parent, if it shows a dominant trait, you know at least one allele. And so could we tell what the second allele is? And sometimes we can, only if we know what their kids are. And so looking at kids can be really, really helpful, because if they had, let's say these two parents had a little b kid, they had a recessive kid, then it can tell, you can work backwards and tell that parent had a little b. Sometimes you can tell, depending upon the kids. Not all the time, but sometimes. So whenever, if you don't know somebody's genotype for sure, you either look at the kids that they have or look at their parents. Because if you're, this is like the example problem I did in class, you did in class, if you have an unknown individual that is, let's say, big B, and we want to know what the second gene is, you look at their kids, and if their kids are little b, then you know they have to have a little b. Or you can look up a generation at their parents, because if their parents had little b, that parent had to give a little b to them. So those are the two directions excuse me, in which you look and you can tell. All right, for the unknown genotype. So I hope you did well on this. If you have any questions on it, we can go over them in class, but hopefully it all makes sense.